My name is Kiara Dooms, and I will be doing a presentation on Paclitaxel, a chemotherapeutic drug. Paclitaxel is also known as Taxol. It was originally isolated from the bark of the Pacific yew tree. It is used to treat many forms of cancer, especially breast cancer. Paclitaxel binds tightly to microtubules and stabilizes them, causing much of the free tubulin within the cells to be locked up in the microtubules. Normally, the growing end of microtubules are protected from rapid depolymerization by a cap of GTP tubulin dimers and other proteins. And GDP tubulin dimers have a slightly different conformation that makes microtubules containing them less stable, favoring depolymerization. Paclitaxel binds directly to the beta tubulin subunits in the tubulin heterodimers. Paclitaxel can help prevent microtubule depolymerization even if the GTP cap disappears by preventing the compaction of the GDP tubulin dimers, which normally causes microtubule catastrophe. This depolymerization is necessary during anaphase. Within meiotic cells, paclitaxel arrests cell division. Cells are unable to proceed from metaphase to anaphase, thereby prevents the cells from proliferating. Paclitaxel interferes with the mitosis stage of the cell cycle. In normal cells, the cell cycle is controlled by a series of signaling pathways by which a cell grows, replicates its DNA, and divides. This pathway includes mechanisms to ensure errors are corrected, and if not, the cell enters apoptosis or programmed cell death. In cancer, as a result of genetic mutations, this regulatory process malfunctions, resulting in uncontrolled cell proliferation. The cell cycle pathway contains 4 phases. 1. The G1 phase, or gap phase, where the cell grows and prepares to synthesize DNA. 2. The S, or synthesis phase, in which the cell synthesizes DNA. 3. The G2 phase, where cells prepare to divide. 4. The M, or mitosis phase, where cell division occurs. The 3 vital checkpoints of this pathway are: 1. The G1S checkpoint, where cells are checked for DNA damage. Cells with intact DNA continue to the S phase. Cells with DNA damage enter apoptosis. 2. The G2 checkpoint. Cells pass if DNA has been replicated successfully. 3. The metaphase checkpoint. Cells pass if all chromosomes are attached to the spindle apparatus. Cells use regulatory proteins and enzymes called kinases to control various steps in the cell cycle. When functioning properly, the cell's regulatory proteins act as the cell's tumor suppressors by controlling cell growth and inducing apoptosis. Mutations causing the malfunction or absence of regulatory proteins at cell cycle checkpoints can result in the cell permanently passing all the checkpoints regardless if they are fit, leading to uncontrolled multiplication of the cells, which leads to carcinogenesis, or tumor development. Paclitaxel interferes with the cell cycle of all cells, not just cancer cells. Because of this, cell cycle arrest occurs in normally functioning, rapidly dividing cells throughout the body, such as peripheral nerves and red and white blood cells. Loss of peripheral nerves leads to peripheral neuropathy, which is characterized by weakness, numbness, and pain, usually in the hands and feet. Loss of red blood cells leads to anemia, where there isn't enough red blood cells to carry oxygen to the body's tissues. This causes fatigue, skin paleness, shortness of breath, and dizziness. Loss of white blood cells, which the body uses to fight infection and other diseases, leads to neutropenia, a condition that increases susceptibility to infection. Can you describe an experiment using common cellular biology techniques that can confirm the effectiveness of paclitaxel at arresting cell division?